Greetings humans, I'm here to talk to you about the cell cycle. So when we're looking at the cell cycle, we're looking at the processes the cell goes through, the phases the cell goes through when it's going to grow and divide. So an organism needs to grow, it needs to make new cells. If an organism needs to repair damaged cells or replace worn out cells, it's going to go through these phases of the cell cycle in order to grow and to then divide the cell into two daughter cells. Before we talk about the cell cycle though, it's important to talk about chromosomes. So when we're looking at chromosomes, we will count the chromosomes by the centromere. The centromere is the center part that holds the chromosomes together. This right here is a duplicated chromosome, while this is an unduplicated chromosome. Chromosome. And it's confusing because this chromosome obviously looks like a lot more. It's because it's duplicated. It's the exact same, identical on both sides, and these are two sister chromatids. What a chromosome is exactly is it's going to be DNA that's super coiled up and tightly packed. The DNA actually wraps around histones. These histones are proteins. It's going to wrap around it. Then the nucleosomes will wrap around itself, and they'll wrap around itself again, and it'll just keep wrapping and wrapping. A nucleosome specifically is the histones and the DNA. So the histones are the protein, and the nucleosome is specifically the histones and the DNA wrapped around. This photo shows the same thing, or this drawing. We have the structure of DNA, and then it's wrapped around these histones, and then histones, this specifically would be a nucleosome. These are the histones. Nucleosome is the DNA plus the histones. They wrap around each other, and eventually condenses into an entire chromosome structure, which is going to be in the nucleus of the cell. All right, so when we're looking at the cell cycle, we have this one really big phase. And this one really big phase is called interphase. This is when the cell's doing normal stuff. It's doing chemical reactions, it's hanging out, it's doing the stuff it needs to do, it's doing whatever the cell's function is, it's doing it. But it's also preparing for division. But this is all the preparation phase. We have gap 1, synthesis, and gap 2. Gap 1 or G1, the cell is going to grow. I always think G for grow. And then it's also going to copy all the organelles in the cell. Synthesis, that word synthesis means to make. It synthesizes or makes or copies or replicates, duplicates the DNA. So this is all about making an exact copy of all the DNA in the cell. And then here in gap 2, we got that G for grow again. It's going to grow. It's also going to make some proteins that are important, and it'll start organizing the cytoplasm, organize the stuff to prepare for this. But the big thing for the gap phases is growth. After we have interphase, this is the whole process. The cell spends most of its time in interphase. Then we're going to have these two division phases. This whole chunk is called the mitotic phase. The mitotic phase is going to include mitosis and cytokinesis. Mitosis divides the nucleus, which is really important because of all that DNA inside the nucleus. And then cytokinesis is going to divide the cytoplasm. Mitosis has 4 phases: PMAT, P-M-A-T, PMAT, and then this divides the nucleus. So this will begin the division of the nucleus, this will end the division of the nucleus, and then cytokinesis will divide the cytoplasm, the organelles, and it'll create 2 new cells. This is what the cell's going to look like during the cell cycle. So this is just looking at the phases. These are what the cell's going to look like at each phase. So interphase, the DNA is still coiled up into that chromatin. Even when the DNA replicates itself, it's still it's chromatin, it's DNA wrapped around the histone proteins, but it's not condensed yet into chromosomes. The nucleus is still fully intact. All the parts inside the cell are doing their part things. And then we're also going to get, what I'm missing from here, is the duplication of the centriole centrosomes. Okay? This is interphase, this is G1, this is S, this is G2, this is all of interphase. It looks pretty boring. Right? Under a microscope, it's going to look pretty boring. You're not going to see the chromosomes. You're not going to see that until the chromosomes condense. chromatin. Here we have the first part of mitosis. So we got mitosis, P-M-A-T, prophase, metaphase, anaphase, telophase. And then this is going to be the division of the nucleus. Prophase, we have the chromosomes condensing. We have the nuclear envelope dissolving. And we have the spindle fibers forming between these two centrosomes. Centrosomes are just two centrioles. I kind of think of centrioles, centrosomes as the same thing, but technically a centrosome is two centrioles and they're producing the spindle fibers. They're forming during prophase. Prophase, metaphase, that word meta, you can think middle, because the chromosomes are lining up in the middle along the equator of the cell. If you think of this as a planet, this would be the equator down the center. The spindle fibers attach to the centromere, the center part of the chromosome that holds the chromosomes together. This spindle fiber attaches, the chromosomes line up along the equator, and the centrioles are on either pole. Then in anaphase, I think A for away, the chromosomes are being pulled away or apart. Now we have this two sister chromatids are being separated to either end of the cell. A for away, so prophase, the prephase, preparing phase, metaphase, middle phase, the chromosomes line up. Anaphase, the chromosomes are pulled apart, the sister chromatids are separated. These two identical halves separated so they can go into each of the future cells. And then we're going to have those chromosomes decondense. They're going to uncoil into chromatin again. The nucleus, the nuclear envelope, is going to reform. So we got two new nuclei forming and telophase. And then those spindle fibers are going to dissolve in this phase as well. The centrioles are still around, but the spindle fibers are going to dissolve. Then after mitosis, P-M-A-T, PMAT, which is the division of nucleus. Now we have the nucleus reform. We're going to get cytokinesis. We're going to see cytokinesis a little different in plant and animal cells. This looks like an animal cell, and then when it pinches off, that's going to be the cleavage furrow, and that's in animal cells. And then in plant cells, the plant cell is actually going to form a cell plate. They can't pinch off because of that cell wall. So the cell plate materials are being brought to the cell wall in vesicles, and then it's going to form that cell plate in between the two new cells. So that would be in plant cells, and that would be in animal cells. So when we're looking at the cell cycle, we're looking at making exact copies of the original parent cell into two identical daughter cells. So if the original parent cell has 46 chromosomes, humans, and our body cells, our somatic cells, we have 46 chromosomes. If we have 46 chromosomes, because we duplicate the DNA in the S phase, after division, each daughter cell is also going to have 46 chromosomes. The number of chromosomes will be maintained because of the DNA duplication, and then we organize the sister chromatids, separate them into two of the new nuclei for the daughter cells. This is making exact copies for growth, for repair in living organisms. The cell cycle is also regulated by something called checkpoints. There's a couple of checkpoints throughout the cell cycle. We have a GAP1 checkpoint. That's going to check a lot of things. This is kind of the do or die checkpoint. So the G1 checkpoint is going to check that the DNA is okay. It's going to check that there's enough nutrients, the cell size is enough. Is this a time for the cell to divide? And if it's not, it's going to stop division. If it gets past the G1 checkpoint, unless there's DNA damage, it's going to go. This is a big decision for the cell. GAP2 checkpoint happens after synthesis. So this is checking to make sure, did this finish? Did we copy all the DNA? And is there DNA damage? Right? Because DNA damage is bad. That's when we can get cancer. And then the other checkpoint is going to be here. And it's going to make sure, obviously DNA damage, but it's going to make sure that those spindle fibers are actually attached to all of the centromeres to make sure that they're all ready to divide for anaphase. So that's going to be the spindle checkpoint in the middle of mitosis between meta and anaphase. When you get uncontrolled cell division, that's typically because you get a mutation in one of the proteins that regulates these checkpoints. So if you damage the DNA that codes for the protein that controls these parts of the cell cycle, then they're not able to control the cell cycle. So you're going to get a cell that divides uncontrollably. When you, especially if this is not working here, that's when you're going to get a lot of uncontrolled cell division, and that's cancer. Cancer is uncontrolled cell division. It can end with tumors, and if it metastasizes, the cancer spreads around the body. Hopefully that was helpful. Please check out my song on the Itsy Bitsy Cell. I'm sorry, the Itsy Splitsy Cell that I wrote myself from the song Itsy Bitsy Spider.